Hi, I'm Charlotte, and I recently finished my PhD using proteomics to study the production of AAV vectors in HEC293 cells. AAVs are adeno-associated viruses, and they get their name from the fact that they rely on co-infection with another virus called an adenovirus. However, AAVs themselves are simple and non-pathogenic, with just a protein capsid and a single-stranded DNA genome. People are interested in AAVs because they can be used as vectors to deliver DNA to cells in a research or therapeutic setting, and I'm particularly interested in how we manufacture AAV vectors for gene therapy. This is usually done through a transient triple transfection, putting 3 plasmids into HEC293 cells and using the cells as a factory. However, this method has a relatively low yield and is unable to meet the demands of the increasing therapeutic market, and has also left AAV vectors as the most expensive drugs on the market. So my PhD has aimed to use proteomics to profile the response of HEC293 cells to AAV production in the hope of finding proteins or pathways that could be used to rationally design the cells to have a higher yield. The first way in which I did this was to use expression proteomics and try and understand whether HEC293 cell proteins change in abundance during AAV production. To do this, I compared non-producing mock control cells to the production of different AAV variants, or serotypes, namely AAV2, 6.2 and 8, and this was done at multiple time points post-transfection. Importantly, the same production protocol was shown to lead to significantly different yields when producing different AAV serotypes, as is shown on the graph here. And interestingly, these serotype-dependent differences in AAV yield were not reflected in the total proteome data. So although the HEC293 cells did show a common response to the production of AAV, as shown in the first 3 volcano plots at each time point, there was no significant differences in the proteome of HEC293 cells producing different AAV serotypes, as is shown in the last 3 volcano plots in each time point. The second approach I used to profile this system was subcellular proteomics, using a method called LOPIT-DC. Here I was aiming to answer the question of whether proteins within the HEC293 cells were undergoing relocation during AAV production. So here on the screen you can see the 2 maps that I generated, one for the mock cells and one for the AAV producing cells. And here we see t-SNE plots with each point representing a single protein, and proteins that have been successfully localized to a single subcellular localization or organelle are shown in their respective color, with the legend shown in the bottom left. By comparing the subcellular proteome maps of mock transfected and AAV-producing cells, I was able to identify candidate proteins that may have moved during this production process. And interestingly, these proteins only had a small overlap with proteins that showed significant changes in their abundance, showing that changes in protein localization and abundance are actually orthogonal regulation methods within the cell during AAV production. Overall, these 2 datasets were the largest proteomic profiling of AAV production to date, and they allowed me to generate a model of the cell during AAV production. Although not discussed here, this led me to identify multiple targets for rational design, which were then followed up through overexpression and shRNA knockdown experiments.